Matt, thank you for the very warm introduction. You've unrealistically set the standard here. It's a real pleasure to be here. I really love participating in summer courses. For the last 5 years, I co-directed with Mark Goldman the Methods in Computational Neuroscience at Woods Hole, and that was a wonderful experience. And so it's really great to be invited to participate in your course here. So thank you very much. As Matt described, our lab is interested in understanding how the brain generates complex sequential behaviors. In particular, learned behaviors that aren't innate. So, for example, in humans, examples of behaviors like this would be speech and language, musical performance, athletic feats, things where our brain has to generate a precise sequence of motor gestures in order for those behaviors to be successful. And on top of that, because those behaviors aren't innate, we have to learn those behaviors through a lot of practice, many thousands of hours of practice. And we also learn those behaviors by imitating others that we see doing those behaviors. So, for example, we learn speech by listening to our parents. So our lab has studied these behaviors using the Songbird as a model system. The specific bird that we study is the zebrafinch. It's a very common bird in the songbird field. Zebrafinches have a characteristic song that they sing that sounds like this, in case you haven't heard it. Okay, each bird has a somewhat distinct song. The song has a very characteristic structure. There's a repeated song motif that you hear lasts about 1 second, so da-da-da-dee, 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 like that. Each motif is composed of 3 to 7 song syllables that last about 100 or 200 milliseconds, and between each syllable is a brief breath that the bird takes so that the bird can continue singing continuously without having to stop. At the finest time scale, song syllables can be composed of individual acoustic elements that can have transitions between them on the order of on timescales of on the order of 10 milliseconds. So there's a very wide range of timescales that this behavior involves. So also singing is learned. A young bird like this is a juvenile zebrafinch. You can see a male bird here in the background that will sing to the juvenile. At an early age, the young bird will listen to the father's song and store a memory of what that song sounds like. That forms the template in the bird's auditory cortex. And then at a later stage, the bird will begin to babble and then, listening to itself, compare its own vocalizations with that of its internal memory and gradually refine its own vocalizations until it can produce a very good copy. It can produce a very good copy of the father's song. And this developmental progression and the requirement for auditory experience and for practice was shown by Mark Kanishi back in the 70s. So birds go through a series of stages. At an early stage, during the learning process, they sing babbling called subsong. Let me just play these for you, actually. So first, the bird will hear a tutor song. Let me play you an example. So this is a bird that went through this learning process. Here's the song that the young bird heard. Okay. About the time he hears that, he starts babbling. So this is a young bird at about 42 days of age. Okay. And then this is highly variable vocalization. The distribution of syllable durations is actually exponential, which is quite interesting, suggesting that there's some very random process going on in generating that vocalization. Then after a few days, you start seeing repeated acoustic structure. after a few more days, you can see that the bird starts developing distinct syllable types. And then by the time the bird is about 80 or 90 days old, he has an adult song that's very stable, it's called crystallized, suggesting its stability. Okay, and then let's compare that with the tutor that the bird heard. Okay, pretty good. So they, okay, so there's a lot of work being done in the songbird field trying to understand many different aspects of this behavior. So an important part of the problem is understanding how the brain circuits involved in this actually produce the vocalization. There's a lot of interest in understanding how the tutor memory is stored in the brain, how the bird generates this random babbling as it's beginning to vocalize, how the comparison of the song that the bird sings is made to the memorized tutor song, to the template, and how the motor system gradually is refined during these several weeks of practice to produce a good copy of the tutor song. So there are a large number of really interesting questions about how brain circuits implement this behavior, and I'm going to focus on just a couple of them today. So I'll describe some work in working to understand how the sequences are actually generated in the brain, and related to that, how those sequences emerge during learning. So it's thought that a lot of the learning process as the bird babbles and compares its song to the tutor memory involves reinforcement learning. There's evidence that there are dopaminergic signals in the brain that indicate when the bird's own vocalization produces a good match to the tutor song, or rather a dip in dopamine signaling when the song doesn't match the tutor song. So reinforcement learning is thought to be a very important process in driving this learning that we see here. But in order for reinforcement learning to work, the brain has to be able to generate a sequential pattern so that you can then use reinforcement mechanisms to refine the vocalization. And so one of the things I'll talk about today is how these neural sequences arise in early learning. And then finally, I want to talk about some tools that we've been working on for analyzing sequences in the brain. Okay, so the brain circuitry involved in song production was largely identified in Fernando Nottebohm's lab in the 70s and 80s. So there are a set of brain areas that are involved in vocal production. The vocal organ called the syrinx is down here, has about 8 muscles on each side of the vocal organ, and it's actually located at the base of the trachea, where there are two, it's a bilaterally symmetric structure with 2 separate sound sources, about 8 muscles on each side. And those muscles are innervated by about 3,000 neurons in the brainstem. Those neurons in the brainstem are innervated by a set of nuclei in what is likely analogous or maybe homologous to cortex in mammals, although there's a lot of debate about the exact relation between the bird's cortex and the mammalian neocortex. Okay, so there are a set of brain areas here. Our story today will focus largely on these two, HVC and RA. There are also feedback loops. This nucleus RA, which is thought to be like layer 5 of motor cortex, projects down to these motor neurons, but it also projects to midbrain structures, which project to the thalamus and back up to HVC. Now, our lab and other labs in the field have spent a lot of time, a lot of effort to record from neurons in these different brain areas to see how the neurons behave during singing, and based on those patterns of activity to develop and then test hypotheses for how this circuit works. So let me show you how those patterns of activity look. So I'll show you recordings in these 2 cortical areas, HVC and RA. The data were taken with a motorized microdrive that allows you to move the electrodes without touching the animal, without handling the animal, and gives much better yield of neurons while the bird is actually singing. So we're trying to record from neurons while the bird is singing. This shows a recent 3D printed version of the Microdrive that's very light. So there's a motor in here, a threaded rod, and then a threaded nut that allows you to move the electrode up and down. Okay, so this shows a recording of a neuron in this Nucleus RA. So these neurons fire spontaneously at about 40 hertz during non-singing. But then when the bird begins to sing, and here you see 3 repetitions of this song motif that lasts about a second. And you can see that this neuron begins to generate a bunch of high-frequency bursts of spikes. You can see that there are about 10 of those bursts during one repetition of the song, and each one of those bursts is reproduced extremely precisely, as Dan Margoliash showed. Dan's lab was the first to record these in singing birds, and he found this incredibly high precision of the spikes in these firing patterns. Okay, and if you record from many of these neurons in the same animal, you can take advantage of the high stereotypy of the song to align the firing patterns of different neurons that were recorded at different times to assemble a population picture of what these neurons look like. And you can see that each neuron generates a sequence of high-frequency bursts. Each neuron looks like it's doing some sort of random pattern of bursts, but that pattern of bursts is precisely reproduced each time the bird sings. These neurons, remember, project down to motor neurons, so these bursts are reaching down and activating muscles in a very precise pattern of activity that generates this vocalization during singing. Okay? All right, so now let's go one synapse upstream from these neurons in RA. So let's look at neurons in HVC. So HVC has multiple neuron types. It has neurons that project down to RA. It also has neurons that project to the basal ganglia that are involved in learning. It also has local interneurons. And we wanted to record in HVC and figure out really what the inputs are to RA that are generating those complex sequence of bursts. So one model that we had thought about is that it's possible that local circuitry within RA is actually generating that sequence of bursts. Now, another possibility is that those bursts in RA are simply being driven by input from upstream, from HVC. And so we were really interested in understanding what those neurons do that are projecting to RA. And here's what they're doing. So this shows a single unit recording. And let me just mention that in order to know that you're recording from the right population of neurons, you have to use some trick to identify them. Here we used antidromic identification to localize the neurons that actually project to RA. So these neurons, this is a recording of a neuron in HVC that projects to RA. These neurons generate a single burst of spikes during each repetition of the song motif. And each one of those bursts is about 6 milliseconds in duration. And if you record from a large number of those neurons, if you record from a bunch of those neurons in the same animal, you can see that each of those neurons is active at a different time in the song. And each of those neurons is active only once. So we then wanted to understand how those bursts are actually distributed throughout time within the song. So we recorded from a very large number of those neurons in singing birds, in this case 40 neurons, 44 neurons, and you can see that those bursts are distributed essentially randomly throughout the duration of the song motif. So, the number of those neurons in HVC is about 20,000, and so we believe that those neurons completely fill time, if you will, in that temporal space of the duration of the song. Okay?
Just a quick clarification question. So notably, what you did not find was an increased density of those neurons firing at the start of a syllable.
That's right. There's a very subtle but statistically significant modulation where there is a slight increase in the density around syllable onsets in adult birds, but it turns out that that modulation is very strong in juvenile birds, okay, and gradually becomes more and more smooth, more evenly distributed in adult birds. So I'll come to that shortly. When we ask the question, how do these sequences emerge? Okay, any questions about that? Okay, yes?
What did you mean by distributed randomly? Because it looks like it's kind of uniformly tiling time.
Yeah, so that's a great question. We don't think they're tiling time. You mean like edge to edge like this. We looked at that. We found no evidence that the bursts were actually tiled in time. But if you look at the variations in density, it's consistent with the variations that you would see if you randomly place bursts in time. Okay, so these observations and other observations have led us to a simple mechanistic model for how song is generated in the songbird. Okay, so the idea is that you have a sequence in time where you have a bunch of neurons. I've drawn these things tiled, even though just because it's easier to see on the screen what it looks like if you tile them. But the idea is that these neurons are active once during the song, and at each moment in time, there is a population of those neurons active. If you look at the average duration of these bursts and the number of neurons in HVC, we estimate that there would be about 200 neurons in HVC that project to RA, co-active at each moment. Okay, so that's the size of that population here. Okay, so the idea is that these neurons, these populations of neurons burst one after the other with some overlap. And what do? We think that those neurons activate downstream in RA a subset of those RA neurons at each moment in the song. Okay? And those neurons then converge down to motor neurons in the brainstem that then activate tension in each one of those 8 muscles in a particular temporal pattern that's very precisely reproduced each time the bird sings, and that's how the bird generates a stereotyped song. Okay, so that's our working hypothesis. It sort of formed the core of the way we think about how the song system works, but we are interested in testing that. There are other ideas about how things work, including ascribing varying degrees of dynamics within this circuit RA or within this or downstream of RA even. So, but anyway, this is how we have thought about this problem. Yes?
What makes possible the synchronization of each? It seems that you have both neurons that are consecutively.
Yeah. So how do you get from here to here?
Yes.
Great. That's the next question. Okay, so there are a number of different ideas about how this works. I'm not going to have time today because I want to get to some of the new tools that we've been developing. But our favorite hypothesis about how this works is that there is a synaptic chain of neurons within HVC, and that's how this thing generates sequences. So that was proposed by Henry Greenside, that you could have neurons connected synaptically like this. It's very closely related to the idea of synfire chains that Moshe Obelist described several decades ago. And the idea is that you have a population of neurons that activates another population of neurons. this model has a number of problems. In principle, you could imagine that if you have a population of neurons and then you have some diffuse projection to another population of neurons, that even if you synchronously activate these neurons, those neurons might have some noise. And then as that propagates, you get some more noise. And then the thing just sort of devolves. Or if the gain isn't set right, then the activity decays. Okay? Or if it's set too high, then the activity blows up. So there are a number of aspects of this circuit that suggest that this chain model might actually work here. One of the particular things is that these neurons are actually intrinsically bursting, so that when you activate them, they robustly generate a burst of spikes. Mike Long, when he was a postdoc in the lab, did experiments strongly suggesting that those neurons generate bursts by L-type calcium channels based on the pharmacology of the bursting process. And in collaboration with Deje Jin, we showed that if you take a synaptically connected chain of neurons and you incorporate that kind of intrinsic bursting property, that the chains become very stable and very robust. So you don't have this problem of too much gain or too little gain or dispersion through the chain. Okay, great question. Yes?
So if you knock out one of those neurons, will the rest of them not?
Yeah, so remember, this is a population of about 200 neurons, we believe. So the other thing is that the neurons that are active at the same time in the song appear to be distributed throughout HVC, which is about 1 millimeter across. So we think that all the neurons that are activating at one moment in the song are sort of spread out all over the place. And it would be very hard to actually, it would be great if they were sort of all in one little stripe within HVC, and we could do exactly that experiment, but we don't know how to do that experiment. Okay, yes?
Are neurons that are coactive during the song also coactive at other points when the bird isn't singing?
So these particular neurons, the neurons in HVC that project down to RA, are actually perfectly silent when the bird is not singing. Okay, so they have some activity during sleep. They have some activity under anesthesia if you play the bird's own song to the bird. But when the bird is awake and not singing, these neurons are just perfectly silent. We've never seen these neurons make a spike when the bird isn't singing. Yes?
Is there anything that differentiates the neurons from each other besides the inputs they receive?
That's a great question. I have always thought that there wouldn't be. Nobody's described any kind of heterogeneity in this population of neurons that project to RA. So I always have thought of them as a homogeneous population of neurons that are connected in this chain. So I would guess no, but it's a fascinating question. Yes?
Isn't this much more consistent with the tiling of time than with the random?
Good. So you might think so, although if you look at models for how these chains develop, I think the answer is that so the way that I've drawn it here is a simplification. If you have 200 neurons in this population and 200 neurons here, they will, some set of these 200 neurons will project. In other words, there will be some connection matrix from this population to the next population, and you can imagine that there's some smear in the exact timing of when those neurons fire. So we don't think that this model strongly predicts tiling. Okay, let me push on. So we don't actually think that circuitry within HVC looks like one long chain like this. We think that our favorite hypothesis right now is that these chains in HVC, this structure in HVC is broken up into distinct modules, each corresponding to one of those syllables. There are a bunch of pieces of evidence that come from a number of labs about, let me just give you an example. So for example, if you flash a light at a bird while he's singing, Jeff Sinks showed that the bird interrupts his song, tends to interrupt the song at the end of a syllable. So there's something particularly sort of fragile about the dynamics of the circuit between syllables. If you stimulate the brain at different times. It supports the idea that there's not one long chain in HVC, but rather short pieces. And our favorite model right now is that there will be a chain of neurons in HVC that generates a syllable, and then the next syllable is reactivated through this loop through the midbrain and thalamus back to HVC. So that's how we think that this thing works right now. So the idea is that HVC somehow develops a short chain of neurons for each syllable that the bird learns to sing. Okay? Okay. All right. So, okay. So let's come back to our description of the stages here. And what I'd like to do is walk through a series of arguments and experiments that suggest a particular model for how HVC develops those syllable models, those syllable chains, those synaptically connected chains, one for each syllable in the song. Okay? All right, so if we're going to ask how those little syllable chains might develop during singing, let's go back and look at the very beginning, the first kinds of songs that the bird sings, this subsong. Okay, so these are highly variable, random syllables, and we could just ask the simple question first: what is it that generates this singing? What parts of the brain are important for generating this babbling vocalization? So we did a number of experiments to work on this, and these were experiments done by Dmitry Aronov, and let me just show you. So the first question we could ask is HVC even necessary for subsong production? So Dmitry did a number of beautiful experiments showing that if you lesion HVC, there's basically no effect on subsong production. HVC is not even required for subsong. Okay? Now that led to the obvious question, well, what if HVC is not required for subsong production, what is it that generates subsong? And it turns out that there's another input to RA from a nucleus called LMAN. That was one of those brain areas that Fernando Notenbaum discovered was involved in the song system by anatomical tracing. And what you find is that, so Dmitry showed that HVC is not necessary for song production, but we wanted to know then, is LMAN necessary for subsong production? Okay, and so Jesse Goldberg, who was a postdoc in the lab at the time, did lesion or inactivation experiments in LMAN in subsong birds and found that when you inactivate LMAN, you get a loss of all of this variability in subsong. Okay, so subsong is normally highly variable, but if you inactivate LMAN, all that variability goes away and you're left with a very simple vocal production. Okay, in this case, a stereotyped song syllable. Okay, does that make sense? So the song at this early age is highly variable, but if you lesion HVC, there's essentially no effect on this variability. The subsong is intact, but if you inactivate LMAN, that variability goes away. Okay, so we think that LMAN is a source of variability that drives subsong, and HVC is a source of sequences that drive adult song. Let me just show you one more thing. If you record from neurons in LMAN during subsong production, you see that neurons in LMAN generate bursts of spikes prior to subsong syllable onsets. Okay? And so we think that what I'm showing here, the red trace shows the spikes, And the black trace shows the song amplitude as these different subsong syllables turn on and turn off, turn on and turn off. And you can see that the L-men neurons are active prior to syllable onsets. Yes?
So the breaks between, I don't know if they're still called syllables, with and without L-man, do you think you had described the gaps previously as breathing? And do you
Yes.
think breathing
Yes.
with L-man, or is it like?
Yeah, so these gaps right here, so the bird is producing a syllable, and then we believe it would be taking a breath between each one of those syllables.
So is that like, do you think it's like an oscillation driving it, or a?
That's right.
Or
So.
just like some other area controlling breathing?
Yeah, so, okay, so let me come back to that question because I think that will be much easier to answer in a minute. Okay, so, all right, so the idea is that we have a variability generator, L-man, that drives subsong very early in development at the time of those very earliest subsong vocalizations. HVC, we believe, is a sequence generator. Both of those areas converge onto this motor output nucleus RA. And early in song, the variability generator dominates, producing these highly variable subsong syllables. Later in development, HVC begins to take over. Sequences emerge in HVC and gradually dominate the production of a sequential song. And a number of studies have shown that L-man generates variability actually throughout the bird's life. In young animals, the variability dominates, we believe, because that variability is driving the vocal exploration required for reinforcement learning. And then gradually that variability decreases until the sequence generator predominates the production of these highly stereotyped song syllables. Okay? Okay, so now I'm realizing that I miscalculated the amount of time that it would take me to get through this. So I'm probably going to sort of go through the highlights of how we think that these sequences for these different syllables emerges during development, and then because I want to get a little bit to the to a method for extracting sequential structure from neuronal spike trains, because it makes a nice connection to what you'll hear about later today, hidden Markov model methods for extracting sequences. So I want to just touch on that as well. Okay, so how do these sequences, how do we go from a case here where HVC isn't even required for singing subsong? We have subsong, but HVC isn't even required for generating these vocalizations, to a case where we have a little sequence for each syllable in the song. Where do those sequences come from? How do they arise? So the first thing I want to show you is what HVC is actually doing during subsong production. So if you lesion HVC, subsong isn't affected, but it turns out that HVC is actually active during subsong. So these are recordings in HVC carried out by, in the lab by Tatsukubo, who's a grad student. And Tats recorded these neurons in HVC and found that in subsong, these neurons that project to RA, actually generate bursts of activity, but those bursts are largely restricted to the syllable onsets. So if you record from one of these neurons, plot when the spikes occur relative to the onsets of syllables, you can see that for this neuron, those neurons are bursting prior to syllable onset. And if you plot the time of that burst for a whole bunch of different neurons recorded in HVC in these young birds, you see that most of them are actually active generate a burst prior to syllable onsets. Okay, so now let's look at HVC, what HVC is doing later on in this, what we call the protosyllable stage, where you start getting these repeated rhythmic protosyllables. So Tots recorded in these protosyllable stage birds, and he found that neurons in HVC that project to RA now become rhythmically active. Okay? And different neurons are active at different times in those protosyllables. So here you can see that this neuron is active early in that song syllable. This neuron is active in the middle of that song syllable, and if you plot the activity of all those different neurons recorded in the same bird, what you find is that those neurons now form a sequence. Okay, but they're rhythmically active. It's a single short sequence. Okay? And if you look at the distribution of when those bursts occur relative to syllable onsets, in young birds, they're right at the syllable onset or before the syllable onset. But as you go to older and older birds, you see that those bursts become more and more distributed in time throughout song syllables. So it looks like something is happening right at syllable onsets early, and then somehow it's growing to generate a sequence that's longer, that fills the syllable. Does that make sense? So we kind of cooked up a simple model for how this might happen. We don't know if this is true. There are a bunch of parts of this that still need to be explored experimentally, but here's how we think about it. So remember, LMAN is generating subsong syllables that projects to RA and generates subsong. Now, RA also has that feedback loop back to HVC, and we think that that projection back to HVC activates those HVC neurons prior to syllable onset. Okay? Now, what does that do? So that would activate HVC prior to syllable onset, but then somehow it seems as though that activity grows, okay? Extends further and further into syllables. Okay? And then we think that the rhythmicity might be produced by closing that loop back to RA. Remember, HVC projects to RA, and we think that that might be a simple model for how you could get a rhythmic sequence in HVC that generates a rhythmically active protosyllable. Okay? And it turns out that that kind of chain growth is very simple to get in a model of HVC that starts with a randomly connected network in HVC. You pick a subset of those neurons to activate. Pick a subset of those neurons to activate them, and by synaptic plasticity, grow a chain. Okay, by a synaptic learning rule that looks a lot like Hebbian learning, can actually grow a chain like this. So let me just show you. So we did some modeling. This was work done by Emily Mickiewicz and Hannah Payne when Hannah was a graduate student. Hannah was a student in the Methods in Computational Neuroscience course at MBL. So here we see that. So you start with a randomly connected network, pick a subset of those neurons to activate, and by implementing a Hebbian learning rule that incorporates some synaptic computation, you can actually grow a chain like this. Okay, and I'm not going to have time to describe, really motivate this very strongly, but we think that the way you go from a single chain to multiple chain is by splitting that single early protosyllable chain. How would you take a chain like this and split it? Well, the idea is that this input to HVC becomes segregated into two separate inputs that differentially activate two different parts of that what we call seed neuron population. And the idea is that then splits this chain into two separate chains that can then lead to the production of two different syllables. So here's what that looks like. So once you grow one chain, if you take that population of seed neurons and activate it differentially, activate it alternately, you can actually split that single chain into two separate chains. Okay? And that model leads to certain predictions about how that population of neurons should evolve. And Tots was able to record in HVC throughout the development of throughout the emergence of new song syllables and found a number of features of the activity of those neurons that matches the predictions of this model. Okay. Okay, so the idea is that you form a single chain that produces the proto-syllable, and then you can take that single chain and split it to produce multiple chains that produce different syllables, and then you could actually take those daughter chains and split them to produce yet more syllables. Okay, so that's the basic picture of how we think that works. Okay, we're very interested in watching these patterns of activity in HVC as the song develops to test this model more thoroughly. The data that we've taken so far in HVC during development is largely neurons reported sequentially one after the other using high impedance from those neurons. The problem is that when you record neurons sequentially, you try to build up a picture of what that network is doing. You're always having to line neurons up to the behavior to try to record from simultaneously. And one very nice way of doing that is by using functional calcium imaging. We've started making a lot of progress on this using the endoscopic microscope, which is unfortunately a little bit heavy for birds, so it limits us to birds that are somewhat older than we want to record from. We've been developing our miniature head-mounted microscope that's lighter and smaller than the endoscopic scope. But data that I'll show you now were all collected with the endoscopic microscope. So the idea is to record neurons in HVC in young birds. We can express CAM6F using viral infection, and this is the kind of signal that you can now observe more than 100 neurons simultaneously during singing, and watch pattern evolves over time as the bird is learning. Okay? Now, here's the problem. Once you take that movie, you have to extract signal from it. So we use methods such as the CNMFE cell extraction method from the Peninsky lab, where you can basically use matrix factorization approach that finds regions in the image that are varying in their intensity, and each one of those little regions of interest in the image produce a time series of activity. And so this is the part we're interested in here. So you have this matrix of fluorescence activity as a function of time for all the different neurons in the data set. Okay, now, I don't know about you, but when I look at that, it looks like a big mess, right? What do you do with a data matrix like that? It's hard. We spent a lot of time trying to cluster. You sort of see a hint of maybe, there are some neurons that are kind of doing similar things, but maybe there's a sequence here. It's really hard to tell. So we tried a number of different methods, and we were unsatisfied with that. So we started thinking about new methods of extracting sequential structure in data like this. And we ended up having a very nice collaboration with Mark Goldman and Alex at UC Davis, and Alex Williams, who's a graduate student at Stanford. Alex and Andrew were students in the Methods in Computational Neuroscience course, and Emily is a graduate student in my lab who carried out the imaging work as well as worked on this extraction method. So I'm going to describe this briefly, but there's sample code here on our GitHub, and there's a bioarchive manuscript that describes this work, okay, as well. So, okay, so let me first, what I want to show you the method in action, so that you can get a sense for what it is that I'm explaining over the next few slides. So this was that data matrix that I showed you a few minutes ago or a few seconds ago, and here's what SysMF does. So the idea is we want to explain this matrix as a set of sequences. Okay, so we imagine that somewhere buried in these data are examples of sequential activity. And we want to find those little bits of sequential activity in the data. Now, there might be more than one. So we're going to extract a sequence. We're going to call it a factor. And if that sequence repeats itself multiple times, then that factor will describe each of those repetitions of the sequence. And if there are multiple sequences, there will be multiple factors. So what you're going to see is on the left, you'll see the different factors that the algorithm is identifying, and along the top, you'll see the time course of each one of those factors. Okay, so here's what that looks like. Okay, so factors by time. Okay, so this is all unsupervised. You just give it the matrix, hit go, and it runs. Okay? See here that it identified 3 different factors and found those sequences in the data. And here what I'm showing you is not a reconstruction of the data. It's literally the data matrix sorted in such a way that the neurons associated with factor 1 are at the top, the neurons associated with factor 2 are in the middle, and the neurons associated with factor 3 are at the bottom. Okay? It's the same data matrix where the neurons are just resorted. And there's the original. And now you can see that these different factors were active at different times in the data, and those were extracted without reference to the song. Remember, we just gave it the data matrix, and you can see that those different factors actually correspond very nicely to non-vocal output. Yes.
You never have a situation in which a single neuron is associated with multiple sequences.
Oh, no, absolutely. So single neurons can be associated with different factors. In this case, that's not apparent, but that happens quite often, and we call those shared neurons. So they're neurons that are shared across different factors. So what you said, a neuron that is associated with different factors is more strongly associated with one factor, and so it gets sorted with the factor that it participates more strongly with. But you would then see that neuron also active a little bit, let's say right there. Okay, so that neuron might have been shared a little bit across factor 2. Yeah, so that was just the demo. So now let me give you a little bit of intuition about how it works, and I'll just keep going so you tell me. Yeah, so, okay, so, but maybe, can I go 5 minutes over? Okay, all right, so, okay, so this method is built off of the idea of non-negative matrix factorization, and our particular contribution to this work is an extension. Let me just explain how it works first, and I'll give proper attribution when it comes up. So non-negative matrix factorization looks like this. You have a data matrix that you're trying to explain, and let's say that there are patterns of activity in the data matrix. So let's say of neurons by time. Let's say you have a group of 3 neurons that are active together at 3 different times in the data matrix. So non-negative matrix factorization works well if the data matrix is non-negative, if it's all positive entries, and you can take, you can describe that pattern of activity in the data as a that has signal at the for those 3 neurons. You take an outer product of that vector that describes the neurons with a vector that describes the in time. Okay, so you can reconstruct that matrix as this vector W outer product with this time vector H. Okay. Only have negative entries as well. Okay, you can also describe matrices that have not just one pattern like this, but also another pattern like that. And another vector W2 and another vector H2, and you describe that part of the data as W2 times H2, and that part of the data as W1 times H1. Okay. All right. And you can write this compactly as just a matrix operation W times H, where W is a matrix that piles all these vectors together, and H is a matrix that piles all those vectors together. Okay, now you can find these W's and H's for any data matrix by doing an optimization, by solving an optimization problem. What you're doing is you're finding a W and H that minimizes the difference between the reconstruction WH and the original data matrix X. Okay, so this isn't. You just take each element in the sequence and square it and sum over all the elements in the data matrix. And Sebastian Sung and Dan Lee found a very nice way of finding the W's and H's by a simple multiplicative update rule that works very robustly. Okay, so now how do we do sequences? So you can also find sequences using non-negative matrix factorization like this. So let's say you have a data matrix that has 3 repetitions of a sequence. Imagine now, instead of a single vector W and a single time course H, we put in a little matrix W here that is like a little template of each one of those repetitions of a sequence. Okay? And this little template has L lags, again, by N neurons. So N neurons by L lags. And now we're going to, in order to reconstruct the data matrix, we're going to convolve that little template with that time course. Right? So if we just take this matrix and convolve it with H, you can see that it's going to stamp a little copy of the sequence there, stamp a copy there, and stamp a copy there. Okay? And that becomes a model for reconstructing the data matrix. If we have more than one sequence in the data, we can include more than one of these W's and H's. Okay? So we reconstruct our data matrix now as this W. Okay, so this W convolved with H, and there's a simple notation, sort of an extension of the matrix multiplication notation. That little star circle snowflake there indicates that we're convolving the W with the H. Okay. All right. So, and again, we can just find W and H that minimizes the difference between the reconstruction W convolve H, I'll just call it WH, and the original data matrix. Okay, and that was a method that was invented by Smaragdus in the early 2000s. Now, you can also fit this using a simple multiplicative update rule, which is just an extension of the Lee and Sung method. Okay. Now, the problem is that this method doesn't work very well on real data. Okay, and it has some limitations that we solved by basically regularizing or constraining this convolutional non-negative matrix factorization approach. So I'm just going to describe very briefly how that method fails, what its limitations are, why it doesn't work that well on neuronal data. I'll just show you how we constrained it or regularized it, and then I'll just give you a couple more examples of how it works. So here's one of the ways that convolutional gives you less than desirable results. So if you have a sequence here, if you're given a data matrix that's got some structure in it, you don't know how many sequences are in the data. Okay, so what you would typically do for NMF or for CNMF is to say, okay, find me 10 sequences. You go fill when it's doing this optimization. And so with these data here, if you say, go find me 2 factors, what it will do is it will just find any 2 factors that reconstruct those data. So here you have a single sequence. But what it does is it finds you one factor that describes the first part of the sequence and another factor that describes the second part of the sequence. You can see that the H's are highly correlated and slightly offset because this H is describing the time at which that early part of the sequence occurs, and that H is describing when the later part of the sequence occurs. And every time you run convolutional map, it gives you a slightly different answer, because there are many different possible solutions with 2 factors that will describe that sequence. Okay? And if you run convolutional MF and measure the consistency of the different results by some metric that goes from 0 to 1, you can see that convolutional NMF gives you solutions that are highly inconsistent with each other. So what you want to do is you want to add a constraint that penalizes correlations like this, where you have 2 factors that are correlated either in W or in H. And you can see here that the H for this factor is highly correlated in time with the H for that factor. And so we can measure that correlation and just add a penalty here. Okay. Yep.
Would eigendecomposition be another solution?
So you can do principal components analysis on a matrix like this, or SVD. The problem is that again, SVD is you're essentially taking a product of a vector in neuron space with a vector in time space, and then you're adding up all those different components, all those different principal components to give you a sequence. But a sequence is not low rank; it's high rank. So if you do SVD, it requires a lot of factors. Whereas this method, if you have one sequence, one factor will be enough. Okay, yes?
You just penalize by the number of factors?
Yeah, so that's another way of doing it. So we've also developed ways of searching through the number of factors, find the one that optimizes some metric. Okay, so it's another way of doing it. This was the first method we came up with, so that's what I'm going to show you right now. Okay, there's a simple way of measuring correlations. H transpose simply shows you the zero lag correlations between the matrix of zero lag correlations between the different H's. And that gives you a scalar that you can add to the optimization problem. Notice that these 2 vectors here are highly correlated, but not at zero lag. So you can solve that problem by putting a little smoothing matrix there, so that when you smooth this and smooth that, they become correlated with each other. So that can work. But that doesn't solve this problem, where it will take a single sequence, explain it as 2 factors, where this factor explains that copy of the sequence and that factor explains that copy of the sequence. All right, the little thing, just you give it a constraint and it does everything it can to get around how you've constrained it. So it'll find any solution that you haven't constrained away. But we solved this problem by basically instead of competing the H with a smooth copy of H, we compete a smooth copy of H with wherever in the data this factor has explanatory power. Okay, so I'm not going to really have time to explain this, but basically this cost function now does a very good job of making these 2 factors look highly correlated. It's really easy to add these cost terms in the denominator of this update rule. And when you do that, what you find is that if your data has 3 sequences in it, the method returns only 3 factors, and the rest are filled with zeros. Okay? It converges very rapidly. It works well even if you have shared neurons in your factors. The consistency score of the resulting factorizations is very high, so it gives you the same solution every time. And we've also developed a method of testing the significance of each one of those factors. So if you take this factor, W, and you convolve it with the data, okay, what you find is that there are times in the data. This is not H now. This is W convolved with X. You find that there are times in the data where that factor has high explanatory power. But if you now randomize the columns in this matrix and do that convolution, what you find is that that randomized factor has very little explanatory power anywhere in the data, but you can now look at the distributions of those W transpose X's and compare it with the distribution of those W transpose X's on the real data, and just looking at the skewness of those distributions, you can find a metric that is very good at telling you when you have a significant sequence in the data that is highly unlikely to have occurred by chance. Okay. We've also tested the effectiveness of this method when the sequences are embedded in different kinds of noise. So for example, you can have sequences in which the neurons participate probabilistically in the underlying sequence. So here you can see that the neuron very rarely is active during the sequence, and here it's active more and more often. And you can see the method degrades at some point. Here we've added additive noise to the sequences. Here we've introduced timing jitter, and here we've introduced temporal warping and quantified exactly how the method fails in the presence of these different types of noise. Just real quick, the method works quite well on very small number of repetitions of the underlying sequence in the data. So we've plotted here the similarity of the extracted factors to the ground truth factors as a function of the number of repetitions of the sequence in the data. And you can see that even if you have 2 or 3 repetitions of the sequence, it does a good job finding the underlying sequence. So it works very well on very small data sets. You have to choose this lambda to run the method, and we've developed a heuristic for finding a lambda that gives a good outcome. And basically, a lambda such that the cost, what we call the correlation cost introduced by correlations between the factors, is balanced by reconstruction error, shown here. So that seems to work reasonably well. Okay, we've tried it on hippocampal data and been able to extract sequential structure in the neural activity of hippocampal neurons, a population of hippocampal neurons recorded in a behavioral test data and also extracted sequences from activity recorded in HVC of singing birds, like I showed you before. Okay, we're slowly getting back to our original task of understanding how these sequences emerge in young birds as they're learning their song. So I should stop there. I've described some of the brain circuitry and mechanisms underlying song production and the emergence of new sequences, and I've touched on some of the new tools that we've been developing for understanding sequential structure in brain circuits. So thank you. So I apologize for going over.
So as a passionate musicologist, I really
Yeah. Yeah.
And that's why I think when creating the matrices between neurons and time, you kind of lose the information about the pitch, right? Because
Oh yeah. So the sequential structure in HPC as far as we know, is not related to the actual sounds that are coming out. It's a timing generator. It generates sequential structure that is the kind of the backbone of how the system puts together a sequence of outputs. But it doesn't tell you what the outputs are.
but the sequences themselves, are they, again, I will repeat that, are they fine-tuned? So they sound together pretty well. So I say I think it was pretty, for me, it looked like each population in the sequence activates like separate tone.
Absolutely.
So the sequence is also to match the tone somehow. And I was wondering if you tried, or is it
Yeah.
or as you say, or your matrices to map them to the pitch of the sequences.
Yeah. So you're talking about the vocal output that occurs at each time. So that transformation from sequence in HPC to vocal output at each time, we believe is happening um, through the matrix, through the connectivity from HPC to RA. And that, we would argue, is where the learning is happening that determines what sound the bird produces at each time. But the sequences in HPC, we think, are independent of sound that's being produced. So the way that I think about it is, if you put it in like the analogy of a player piano, HVC is like the clock that rotates the drum. What sound actually comes out is determined by where the holes are on the piece of paper, right? It's separate from the clock.
So the entire mystery being what the information comes back.
Yeah, so there's a whole separate topic about how that learning is carried out, and that's known to involve basal ganglia circuitry, and basically it's thought to be a reinforcement learning mechanism. Yeah. Chris.
Question about one of your first slides described an early sensory stage and then a formation of a template, and all the learning that takes place subsequently is so I wasn't completely clear on this. So,
Yeah, that's because I didn't describe it, and I'm not totally clear on it either. So.
So I guess my question is basically, imagine that you totally prevent the bird from any exposure to parents' song after the initial exposure phase. Have any effect on the development of learning of the song? And I want to kind of contrast that with, or compare that with the case of children learning language. If you have kids and they become deaf at age 15 months,
Mm-hmm.
Is there any evidence of them kind of acquiring templates that they can carefully store, and then later when they're trying to, I guess there's a problem because they can't hear their own speech.
Yeah. So you've raised a number of really important issues. So one is the question of what happens to the song if it's not exposed to the parents. And another question is what happens if it can't hear itself anymore. And each one of those has a different sort of characteristic result. So if you take a young bird and you never expose it to the parent's song, it develops an abnormal vocalization called an isolate song. Okay? And that has consequences in the structure of the neural circuits and the firing patterns, and we've been looking at that question. You can also take a bird and expose it to the parent song and then take it away from the parents so it can't hear the parents anymore. Birds seem to be able to store a template of the parent's song after a small number of exposures to the parent's song. So it doesn't need exposure to the parent, to the father, during the whole learning process once it's formed that template. Okay? And now, if you deafen a bird after it's learned it anywhere as a juvenile, the song very rapidly degrades. If you deafen an older adult bird, the song still degrades, but the older the bird is, the longer it takes the song to degrade. So I tried to encapsulate.
So I mean, so again, I don't know if you know, but remove children from access to any feedback other than their own. So you give them initial exposure, and then you remove them. They only have their own feedback.
Mm-hmm. Their own. They can hear their own voice, but nobody's saying, "Oh, that's good."
Nobody gives them any more exposure. They're silent for the first 8 months of their lives, but they have a lot of exposure, and then afterwards they can talk and hear themselves, but they can't hear anybody else.
Yeah.
Just wondering if that experiment or any version of that has ever existed.
Yeah. So in birds, if you expose the young bird to the tutor song, you can then take him away from the parents, and he can still learn a good copy of the song.
One or two more questions, and we'll take a long break.
So, I have two quick questions. The first one is, so potentially the sequential element could be trained on the parents, and then you could generate an encoding matrix for the children that are learning from the parents.
Oh, that's
Because
interesting.
the structure should be similar. They're learning the same structure.
Mm-hmm.
And in general, is it possible to use this method to generate encoding patterns for, especially for species that learn multiple songs in their lives? So to predict, based on what we know from some of them can learn 20 different songs. So like based on the songs that we know they've learned, can you generate encoding patterns for a new song?
Mm-hmm.
So that's the first question.
I'm failing on your first question, so I don't know. I think that, so first of all, it sounds like what you're proposing is you would record a bunch of neurons in the father's HVC, and then record a bunch of neurons in the juvenile's HVC, and I would guess that you would find a sequence in the father's song, and then you could map that to the sequence in the juvenile song. Yeah.
So in a sense, can you predict the structure, what's going to look like?
So, okay, so now you're asking whether you can predict like the syllable structure, and I would say that you might be able to predict aspects of the timing of the song, not of any other level of detail.
Like an encoding model that I would know what I should see and then go and see what neurons are showing.
You would know what the duration of the song might be. The
Sequences. So I know that there are certain neurons that should behave like this in order to keep the timing of this.
yeah.
So I go and look I use that encoding model
Yep.
to see whether that.
Yeah. Could work.
The other
Could be
question is,
interesting.
so a lot of species, they're females, they can perceive and they judge the song and
Yep.
similarity of their father's song, but they don't sing themselves. So they should have some sequence perception, but not production.
So, yeah, great question. We have no idea how that works. It's really interesting. Because I think that's closely related to the mechanisms by which the juvenile male stores the template. So, because he has the same sort of preferences for species typical song. So it's a really interesting question how a young bird would have a preference for its own species song when it's never heard its own species song. So suggesting there could be genetic mechanisms that set up a structure that's particularly receptive to a kind of song.
But there are some birds who imitate other species songs too.
Yeah, so most birds will imitate another species song, but they will always prefer to store, if given the choice, they will store a memory of species typical of their own species song rather than another species. But if they're only given the other species song, then they will store that. It's really interesting.
Last question, and we'll take a break.
Yeah.
So, the bad links in which the random generator
Yeah.
predominates,
Yes.
that shifts over to the clock through development.
Yes, gradually. Yeah.
Does the have some functional relevance even after the clock?
Yeah. So Michael Brainerd's lab has studied the role of this variability in adult birds, and in particular, he and Alison Dope have found that there's a very strong social modulation of the variability. So when the bird is singing to a female, the variability is very reduced, And so it's like a really good version of his song that's very clean and low noise. But when he sings by himself, L-Man sort of turns on and makes the song more variable. So it's been interpreted that there's a practice mode and a performance mode. Yeah.